Hi everyone, thank you very much for the invitation to speak. My name is Lyndon Parks and I'm a postdoc at the University of Pennsylvania. I'm going to talk to you today about a project I worked on linking transdiagnostic dimensions of psychopathology with deviations from normative neurodevelopment of brain structure. I apologize I couldn't be there to present this in person today, but I've listed my contact details at the bottom of the slide, through which I'm happy to take questions from anyone who might be interested. I'm going to start today by illustrating the assumption of the case control design, which has been a cornerstone method of our attempts in neuroscience to map brain abnormalities associated with mental disorders. This design assumes that patients are cleanly separable in terms of their symptoms and their underlying biology, and that this separability should aid in differential diagnosis. Now, while we've certainly learned a lot about the link between disorders in the brain using this approach, as a field we've kind of come to terms now with the fact that this assumption of separability doesn't really hold in our data, not at the level of symptoms or the biology. And of course, when we consider how we diagnose patients, this lack of separability is not so surprising. And we know that patients can be diagnosed with a certain disorder in many ways, which leads to groups of patients having very high levels of symptom heterogeneity despite all being assigned the same label. And in turn, it's not so surprising that patients who look dissimilar clinically also exhibit biological heterogeneity. And there's a lot of research in the field that supports that idea. So this suggests to me, as well as to many others in the field, that our reality is likely much more dimensional than it is categorical. A great example of this is something like the study of the psychosis spectrum, which shows us that psychosis symptoms are not exclusively present in disorders like schizophrenia, but are also present to sort of varying extents in the broader population. In turn, this dimensional perspective says that while the extreme expression of psychosis symptoms might underpin disorders like schizophrenia, studying the full spectrum and that spectrum's link back to the brain may serve to help improve our understanding of pathophysiology. Another layer to this is that this dimensional perspective doesn't occur in a vacuum, of course, but rather any specific dimension, like psychosis, is likely part of a broader transdiagnostic space, and that assumes that multiple symptom dimensions, as well as dimensions of cognition and developmental processes, all interact and are likely underpinned by multiple interacting biological signatures. So a central question becomes, how do we approach this space, this transdiagnostic space, to find more clinically useful biomarkers? One approach that has gained some traction on this problem is the p-factor hypothesis. The p-factor hypothesis states that variance in psychiatric symptoms may be best summarized into a single factor first that explains individual variation in all forms of psychopathology, a sort of total symptom load, if you like. And this is kind of analogous to the idea of the G factor from general intelligence. Once the p-factor or the general factor is accounted for, specific factors can then be derived from the residual variance in the data that measure specific dimensions of psychopathology that are all orthogonal to each other as well as orthogonal to the p-factor. And that's what's being illustrated here on the right. So using a p-factor model, we wanted to ask the following questions. We wanted to ask, do dimensions of psychopathology from the p-factor model explain deviations from normative neurodevelopment? Does the p-factor explain structural brain abnormalities that are commonly reported in case control literature? And can we use the p-factor model, the p-factor, as a nuisance regressor to explain overlap observed in case control analyses across clinically dissimilar disorders? So to address these questions, we took approximately 1,300 individuals from the Philadelphia neurodevelopmental cohort. In every participant, we assessed psychiatric symptoms using a broad battery of 117 items that index symptoms across disorders that span all major axes of psychiatry. Using these data, we estimate a bifactor model where overall psychopathology was parsed out first, and then specific dimensions were estimated that are listed here on the slide. We then combine these data with estimates of cortical grey matter volume in each of 400 brain regions. And then we combine all of those data using an approach called normative modeling. Essentially, for each region, what we do is we estimate a normative model of grey matter volume development. where age-related changes in volume were estimated using 410 healthy people in our sample, and then we estimated deviations from those trajectories in the 900 or so remaining individuals who experienced varying degrees of psychopathology. For sake of time, I'm not going to cover this approach of normative modeling in great detail, except to say that this approach differs from simply residualizing brain features using something like least squares regression, which is pictured at the top here. The approach we take is to use a Gaussian process regression that allows us to not only estimate the nonlinear age trajectories for cortical volume in our sample, but also to explicitly model and incorporate into our deviations estimates of normative variance in the age trajectory from the healthy data and predictive uncertainty from the model, which is illustrated here at the bottom. So using this approach, what we do is we estimate deviations from normative development of cortical volume, and then at each region, we estimate the correlation between our dimensions of psychopathology and these deviations to examine to what extent our dimensions explain abnormalities in normative neurodevelopment. Now, as you might expect, we find that in our data, cortical volume reduces globally as a function of age for both males and females, which is what I'm showing on the right. This means that for the rest of the results, we interpret negative deviations as kind of an advance on this normative reduction of cortical volume, and positive deviations as a delay in that normative process of reducing volume. So, concerning correlations between deviations and psychopathology, we find that overall psychopathology showed the greatest number of significant effects, which is all this plot is showing here on the left. The plot on the right, though, shows that these effects were all negative for overall psychopathology, indicating that greater overall psychopathology was associated with widespread neurodevelopmentally advanced reductions in grey matter volume. Critically, the regions I'm highlighting here in the red circles, so the inferior temporal cortex, the dorsal anterior cingulate and posterior cingulate, and the medial OFC, are regions that are kind of commonly reported to show structural abnormalities in a broad range of disorders under case control designs. This is including disorders like anxiety, depression, schizophrenia, bipolar disorder, and others. So if we now look at some of the specific dimensions that are relevant to those disorders I just mentioned, such as our psychosis and our anxiety dimensions, we can see that these regions are less implicated. For example, neither psychosis or the anxiety dimension implicate the medial OFC. Instead, psychosis implicates more of the lateral OFC. The cingulate is implicated much less, or to much less of an extent, in each of these phenotypes. And the inferior temporal cortex is primarily implicated by positive psychosis symptoms. So what we find, or what this is showing us, is that these regions that are commonly implicated in the case control literature are actually most extensively associated with overall psychopathology in our data. And so this suggests that the p-factor model is useful for teasing apart brain signatures that are specific to certain dimensions of psychopathology from those that may be more disorder general. So we also tested for differences in effect sizes between overall psychopathology and specific factors within those a priori regions of interest. So using a bootstrapping approach, here we found that in each of those regions I mentioned earlier, the effect size of the correlations for overall psychopathology were stronger, and often significantly so, than the comparable correlations with the specific factors. Next, to kind of test this idea and dig in a little further, we selected 136 individuals with clinically significant depression and 163 individuals with clinically significant ADHD, two disorders that have quite different clinical presentations. Note here that patients in one group actually did not have comorbid diagnoses of the other. Then, in each of those two groups, we separately calculated the regional Cohen's-D's value, comparing deviations from patients to deviations with matched healthy controls. And these two healthy control cohorts were also independent. So in other words, what we've done is we've simulated two totally independent case control studies in two different disorders. And then what we do is we repeat that whole process with and without controlling for overall psychopathology as a nuisance covariate. And the results are shown here on the left. So what you can see is that when we don't control for overall psychopathology, which is the green distributions, both groups show negative effects, demonstrating on average a greater level of negative deviations for cortical volume compared to their respective healthy control groups. This effect shifts significantly towards zero when we add in overall psychopathology as a covariate, and that's what the orange distributions are showing. This happens for both groups, though it is clearly much more pronounced for depression, which is on the bottom left. More importantly, though, the plots B and C here show the spatial correlation between Cohen's D's values across the two groups. So here, each point in plot B and C is a brain region, not an individual, and we can see that the group level effects, these Cohen's D's values, of these two clinically dissimilar disorders correlate at about 0.15. So there's some spatial overlap occurring when we don't control for overall psychopathology in subplot B. Critically, this correlation actually reduces down to 0.08 and becomes non-significant when we add in that covariate for overall psychopathology. And so this suggests that the overlap we observe between these two groups that are dissimilar and don't have comorbidities for each other is explained partly by this factor, overall psychopathology. So to sum up, overall psychopathology is associated with deviations from normative grey matter development in areas commonly reported in the case control literature. Specific psychopathology dimensions yield sort of spatially varied patterns of abnormal grey matter development, and case control comparisons for depression and ADHD appear to overlap spatially, and this is explained by, to some extent, by the overall psychopathology factor. Now, of course, there's always more to a project than can be covered in a short talk, so if you'd like to learn some more, I suggest you check out our paper in Translational Psychiatry on this project. But for now, I want to thank all my wonderful mentors, collaborators, colleagues, and of course, thanks to my funding sources, and thank you all for listening.